5 से चालू कर रहा हूँ, 4 लोग ज्वाइन हुए अभी। यूट्यूब पे चल रहा है ना लाइव। हाँ, ये लाइव जो ऊपर लाइव चलेगा ना, तो यहाँ रिकॉर्ड नहीं करता ना, डायरेक्ट यूट्यूब। डबल खर्चा क्यों? कैसे करते हैं वो? मतलब मुझे नहीं पता कि तो तुम मेरा चैनल को सब्सक्राइब भी नहीं किए। जब मैं तो कुछ बात करूँगा, क्यों बताऊँ तुझे? किया है मैंने। नहीं किया है। नहीं किया? मैं कर रही थी। मैं अंदर बैठी हूँ। हाँ। So hello all. So let us start for this week 4's live session. So welcome you all in this week 4 live session for the course Introduction to Proteogenomics. So I am audible, right? And my slides are also visible. Okay. Yeah, so in this week mainly as you all have gone through the lecture, so mostly it has been the proteomics data analysis and the techniques that after the raw files are generated, that is your YINs and BINs are generated, and after that initial software analysis, the proteins that you have got in different, if you are studying in different conditions or in different treatment conditions or any two groups, so the different proteomics proteins data that has come that needs to be analyzed. For that there are different techniques for the, and the data that comes out contains many technical variations or biasness, and we need to remove all those to get the proteins or the biomarkers that are actually involved in the role in the disease and is biologically significant or is in or is changed or differentially expressed due to the biological reasons and not the technical reasons. So for the case study for today, here another this of the paper of the CPTAC paper of the proteogenomic analysis of human colon cancer. If we look at this paper, that they have used, apart from the genomics, mutation, copy number alteration, transcriptomics, they have also done proteomics and phosphoproteomics. So here we will look at how they have done the proteomics data analysis, so that you can appreciate that the why the different techniques that have been discussed in the lecture, that how that is used daily worldwide to for the analysis for the identification of these biomarkers and reporting those values. So here if you go and see their paper and in the methods, so here they have done, you can see that LCMS for the level free proteomics data analysis and also for the TMT level data analysis. So let us, today we will focus mostly on the level free quantification, level free data analysis. So if you go and search the read this paper, so here you will find out the level free proteomics data analysis. So here you can see that the first the raw MS data. So here there are different approaches you can take, I think, which will be discussed more in the later on in the other weeks. So the raw MS data that is generated, now you need to identify, because that contains the chromatogram the of the different peaks. Now you have to need to identify those peptides and finally the proteins and their intensities. Then only you will be able to compare between your control or the diseased or different groups, that what are the proteins or the markers that are getting differentially regulated, and finally what are the pathways or what are those involved in those diseases. Now if you so here you can use different techniques. So they have used that, they have converted to MGF and MGML files. So MGML is one of the freely accessible, means at format, data format, so raw, so that can get accessed by different software. So using that, so here now you will understand that they need a protein database. So for the identification of the peptides, so this generally needs a database, and that's where the proteogenomics approach comes into play, where you can uh, provide your customized database to identify those mutated peptides. So here for the any proteomics data analysis, you need a robust protein database from where the peptides, so that it will match with your raw data and it will lead you to identification of those protein sequences. So now if you all remember that in previous previous thing, that in the mascot we used to keep parameters for the carbamidomethylation was specified as a fixed modification. So here also in this software also, for the carbamidomethyl is been kept as a fixed modification, and the oxidation of methionine has is and along with the deamidated NQ was kept as a variable modification, and then the different parameters were given for the MSMS scans and all, and finally the uh, proteins were identified. So now when the proteins were identified, then after that they used a quantile normalization. Now if you have gone through the lecture, so you know that there are different kinds of normalization methods we can use. That is one, the mean centering or the median normalization or the quantile normalization, and every normalization has its pros and cons, and it no such normalization technique can be robust, but it depends on the user and to how to use or to what are the things, what objectives behind the uh, using those uh, using those techniques for normalizing his or her data. So this is done, then the normalized and then log to transformed, and then further taken forward for the downstream analysis. So here you can see there I have kept some plots. So this plot is known as a volcano plot, where you can see that there are some of the proteins that are the significant upregulated proteins, and there are some proteins that has been told that significant downregulated proteins. So what does a volcano plot represents? Volcano plots represents both your p value, your significant values that with the t-test or any statistical test that you performed, and in the and also the fold change difference when you do the log transform and find that what are the ratio of the disease versus control, that it is upregulated in this disease a particular protein or downregulated in disease. So you give a specific cutoff. For instance, for log to fold change you will give like a two-fold cutoff, or a 1.5 cutoff. So log to see two-fold cutoff means this particular protein is 4 times upregulated in the disease state, and if it is other way around, -2, then means it is down regulated in the disease state. So in this way the volcano plot represents. So from the data that we get after running in the mass spec, we get the peptides, we identify the proteins, then with the after that the statistical analysis finally helps us in understanding or determining the proteins that are significantly dysregulated. And also the here another is that the median proteome difference. So if you can see that the control tumor versus normal, here the what is the median difference. So mostly the proteins will remain same, but a few proteins that will be away from the distribution in the range of away from 2 or 3 standard deviations, and that those proteins are significantly dysregulated. And this Venn diagram shows the different proteomes and the phosphoproteomes among the total proteins. So here does anyone have any question or anything to ask? Please reply, then I'll move ahead accordingly. So I'm guessing no. So yeah. So now another, I would like to give another case study of the paper where it has been used, quantitative proteomics study, where only proteomics study has been used to identify the malaria severity and complexity. So you know that the disease like malaria can progress from non-severe to severe cases. So now the question comes that why in certain patients it is showing non-severe and why in certain patients it is showing severe. For the cancer and all tumors and all, you can say that okay, that this patient is has some other genetic modifications or alterations, but here also you can see that there are certain cohort of patients that are becoming severe and that are coming non-severe due to the difference or alteration of the proteomes or the particular pathways that leads to progression of a disease and varies from a person to person. So here this study has performed a quantitative proteomics study to identify the why malaria severity and complexity. So here you can see that the severe falciparum malaria and the non-severe malaria, it has also again performed a volcano plot, and you can see that there are different proteins that have been found to be regulated, upregulated and downregulated in both the cases. And this is one of PLHD plot where this healthy controls and the severe and the non-severe are shown to be differentially clustered together, or in that there is a difference in the cluster. That means that there are certain proteins, certain mark that are differentiating the non-severe but with the severe ones. So there are certain proteins that are getting dysregulated and leading to cause for the person or the patient to become more severe. So here how they are done is that here it was a labeled data. So after performing the from the software identified the protein, so here run to run normalization using a reference pool. So here a reference pool was used to perform run to run normalization. So I'm just giving you the examples that there are different techniques through which is done, but all the techniques you need to be carefully understand and apply accordingly in your data, and all these are very much in use over the all over the world too, and are being used in a regular basis for the identification of different biomarkers. And 66 proteins were selected, so adjusted p-value. So again in the statistical t-test you understand that what is the difference between the raw p-value and the adjusted p-value. Why the adjusted p-value is generally used so that the false discovery rate gets much more reduced, and the biomarkers that we get or derived, we can say that yes, we are confident of it, that these are the result of the biological variation, these are causing the disease progression or the diseases, and this can be taken forward. So since again there are missing values associated, so the mass spectrometry data, if you do, because of the various technical complexities, so one peptides get identified in one sample and the same peptide doesn't get identified in the same sample, or there is a differences in the intensity level, and there is also limit of detection, limit of quantification, so all those are detected, doesn't get quantified in the mass spec, and also the software cannot quantify those. So taking all those consideration, there are certain considerable amount of missing values comes in proteomics data, so we need to impute those data, if because otherwise uh, majority many biomarkers we can lose which are of our interest, which can be of genuinely interesting proteins. So there with a certain percentage of the proteins, we allow the other we throw away those proteins and allow the other proteins to be uh, impute missingly, means impute it, so that we can take and perform the further analysis. Also I think by the lecture you can understand that there are three types of missing values, so which are missing completely at random, or missing at random, or missing not at random. So which are missing completely at random, so we are good to go uh, to trace those data and go ahead with the other data present, but which are not missing not at random, so we cannot just throw away them, and we have to look at what certain percentage of samples are showing those data, and then we need to impute those, and then normalization is needed to perform, so that all the samples are on the same scale to differentiate between, to find identify the differences between the control and the altered state. So these are the steps that have been followed in this study and this paper also. So now another thing I thought to keep it is that, as you all know that how this COVID-19 and pandemic has shown that problem, and the world has faced so much difficulties, but the proteomics data can here also reveal, and also you know that during COVID, especially during the second wave, that there was a huge crunch of hospital beds and spaces, there was need to identify and understand that which patients are going. Still now there is it's still happening, though in a much lower rate, but still there is a need to understand that which patients will develop the severity and which patients will not, so that the patients that will, if we can predict those patients that they are going to be severe in coming days, then those patients can be taken of special care and can provide the hospital treatments well ahead of time. So here also proteomics can reveal those prognostic markers which can help in understanding those disease severity. So again here, if you can see the different uh, so if you go through and the how the steps have been performed, so here you can see that the raw data sets were processed with one of the software and using a database. So here by these studies you can well appreciate the need of the database in identifying and in proteins in the proteomic role of these databases in the proteomics studies, and then with different LFQ parameters, and afterwards then a sample wise correlation analysis. So another way of, because the it is very much important to understand the quality of the data generated, because it might happen that in certain, during certain one sample run, sometimes some sample has been, something has happened in the instrument, and some samples did not show the same coverage or something, then you need to perform a sample wise correlation analysis to understand the data quality. And then you, for instance here 74 samples were there to understand the correlation analysis, but 3 samples did not meet the standards, so then these 3 samples were discarded. Now if you take these 3 samples and go ahead with the analysis, because of these three samples your data will get skewed to one place and you can lose uh, interesting signals. So the samples can be of different issues can be there during the sample preparation or during the run in the instrument. So all this can very much affect your data and further downstream analysis. So you need to have a very robust data quality check. So for instance, in genomics data, firstly after you get the raw data, you do a QC check using first QC, as if you are remembered, and you check the Q30 score, that how much that the data is correct, its coverage and all. So in the proteomics data also this is done to check the different data quality check is done to identify that the quality data is of the highest quality standards. Then the missing values were imputed in the k nearest neighbor algorithm. So what k nearest neighbor algorithm has been told in the lecture, that there it checks on the samples that which are nearer to it, that how much is the value, and by that it imputes it using a machine learning algorithm. So here you can see that there is a software called Metabo Analyst. So we will be doing hands-on today completely with the Metabo Analyst. So you can see that with using those software, how with these data sets, how one can solve or one can predict different problems, different diseases markers, and that is being used daily. So you will also be able to uh, make these kinds of plots and can analyze this kind of data. So after that, the differentially expressed proteins were determined using t-test, and then some p-values less than 0.05 were taken, and here the t-test independent samples were taken with the Bonferroni correction. So if you remember that Bonferroni correction is the one which is the most conservative kind of correction, and it takes that the samples are independent, and here it calculates the p-value, whereas the Benjamini-Hodgberg correction is used when you know that the data sets are dependent with each other and they are not independent. So here where you perform the Benjamini-Hodgberg correction. So after that, further the clustering and the PC and the PLSTA plots have been done to identify. So the basic thing is that all the papers you go through the proteomics, I have just given 2 3 examples which has helped in understanding different infectious diseases like malaria or COVID, and those one kind of CP type paper. But mostly the workflow is followed is that after you get the data set, you try to identify the percentage of missing values, do the missing value imputation, do a log transformation, do a normalization, and then do a statistical test to identify the differentially regulated proteins. So this is the basic common workflow that is produced. And then here comes in between that if the samples are run in between batches, then you need a batch correction method using Combat or limma packages, or if there are something differences in the normalization technique, you need to have a very close look on which kind of normalization it is going and how the distributions are coming, and then finally proceed for the analysis. And also you need to have keep in mind that you need a t-test or a moderated t-test based on the basis of the number of samples, that if you have a greater number of samples, then your mean and variance will be much more robust and you will get a good quality amount of data. But if not so, then you need to provide different values, and be based on the basis of the number of samples and on the basis of the data quality, you need to adjust your p-values, that you will go with the conservative uh, correction, or you will take the raw p-value or the FDR value. Again, you need to be careful because the raw p-value may contain some false positives also. So these are all dependent on the analysis, and this is how you proceed with it. So here you can see that this is again a volcano plot, and the different markers. So this is a non-severe condition, severe condition. So you can see that this amount of proteins have been identified. So you can clearly see that there there is a difference, like this is upregulated in the non-severe and it is downregulated in the severe. So this protein might be has some effect which helps in body to have a immune effect or something that needs to be looked at. I'm just giving an example, but this is getting down regulated and this is the patients are showing a severe condition. Similarly for the ApoA2 or the ApoB SERPIN-D1, so you can clearly see that that in one cohort it is upregulated and in one cohort it is downregulated by this heat map. So you can generate this type of graphs, you can understand those types of proteins and can further take forward for the different biological analysis. So again, this is a PLHA plot and this is a volcano plot. So now we will go and see a very user friendly software meta-analysis. So there are different kinds of software which helps you do the secondary analysis. So you have Perseus for the max current output file that is being widely used. You have Protege which you can use. There is MStats which you can use. There are numerous software. So we will be using one freely available web tool, MetaboAnalyst, to perform this kind of analysis. MetaboAnalyst is generally done to identify metabolomics data, mass spec based metabolomics data. But since the proteomics data is also quite kind of similar, so we can go ahead with it, and since it is user friendly and freely available, we can use. And also you have seen that the MetaboAnalyst has been used in proteomic studies and has been published also. So it's not an so we can go ahead and use this software to identify different to analyze these data sets. Okay, so I'm pasting these two links, so I'll request you to go to it and also use these data sets to follow the steps what I am doing, and we'll do it side by side so that so sorry, I didn't check the chat. So Jasera, can you explain fixed and variable modification? So fixed modification, I had explained it in the previous session that when we perform a tryptic digestion, we need to reduce the proteins from their proteins. Cysteine residues have different disulfide bonds, so we need to reduce those to using TCP or DTT. And if you go through any proteomics this paper's sample preparation, so you can check that the proteins are reduced first so that the trypsin can act on the proteins. So in the on during that time, so there a modification called carboamidomethylation occurs, means happens at the cysteine residues. So when we perform a data analysis, we need to provide that cysteine carbamidomethylation as a fixed modifications, and the variable modifications like oxidation or the phosphorylation at STY site, that depends on the protein to protein and the peptide to peptide. So if we give as variable modification, then the software will look that if the charge of the mass of the oxidation state or the phosphorylation state happens occurs, then it will consider those peptide as oxidized at that level. So these are the fixed and variable modifications. So do you have any other did you understand, Jaseda? Okay, so I have pasted the links of the MetaboAnalyst, so everyone just go to it, click and go to it. Let me know that when you all are ready. So I am the dataset link. Let me know that you can access this dataset or not. Sure, will let us do this, then we will come back to that. Once you have opened MetaBoAnalyst and have accessed this, just go and access this dataset and download this dataset and let me know. Just write in the chat when you all have opened it, okay? Okay, fine. So everyone is in this, so everyone got this page, right? So if you go, so I am also keep on pasting the different. There is a MetaBoAnalyst this page, right? So now we click here to start, and you will get this kind of page where you will get LCMS spectra processing. So you can see that raw spectra, MZML, MZXML. So MZML, XML are one of the formats of the raw spectra that gets readable by different readable by different software. Then the intensity peaks, enrichment analysis, statistical analysis. So where we will go? We will go to the statistical analysis one factor. Okay, and you can see that this kind of page. Okay, so everyone is on the same page. Yeah, okay. And now is that we will select the peak intensities. So can anyone tell what is the meaning of this peak intensities? So what are those? What do you mean by these peak intensities? Write anything, yes or no, that you know or don't know. Okay, so peak intensities here refers to the protein that the once the proteins that have been detected in LFQ, the level free quantification. So what are those intensities? So peak areas of those proteins in that has been identified based on the peptide and their intensities identification. So LFQ intensity is given for each protein. So here that is the meaning of those. So here it refers to the now the data sets. I'll just open the data set. So you all just download those data sets. So you all can check that this is the dataset format where you have the control. So you have 7 control and the 7 diseases, right? And you have, we have mentioned it as 0 and 1, and these are the different protein IDs, and these are their intensities across the control and disease. So this is the data set. So I think you all are clear with the data set. There is no problem in understanding what kind of data is, right? So once you have opened it, now we will, what we are taking it as that these are not paired samples. So you all know what is the meaning of paired and unpaired samples. So any one of you, if you have done a statistics class, paired t-test and unpaired t-test, when you perform paired t-test and when you perform unpaired t-test, any one of you? You all know any? Just write na yes or no that you know or don't know. Other than Urvi, Jasera, or anyone, not not samples that are dependent. So if if let you let us take a, if you take a mice, okay, if you take a mice, and in that mice you first take the control samples. Let us assume that the mice has some problem, like the disease condition, for any neurodegenerative disease or any kind of disease. So that mice, you take the data, take the sample and collect the data, and after that you treat that mice. Okay, you treat that mice with different drugs and all, and then take the data. So then the same subject has been, data is from the same subject but two different conditions. Then you generally take it as a paired test. So when you perform on the same subject, but when it is different from the, when you there is two different mice, one is a controlled and one is a diseased, then you will take, then you cannot take paired t-test. The paired, we cannot perform paired, I cannot say the samples are paired, the samples are unpaired. So if the same patient you take control samples as well as the tumor samples, then the samples are paired. But if the samples are different, means different patients, you are taking different samples, then the samples are unpaired. So am I clear with the paired and the unpaired difference? Okay, so now we will, assuming that the samples are unpaired, so we will say that samples in columns unpaired. Why we kept samples in columns? So you can see that the data structure is like that. There is a sample 1, sample 2, sample 3, control 1, control 2, control 3. So again, you all understand there are these terminology that there you can, you will hear terminology like the replicates. So there are two kinds of replicates. One is the technical replicates and one is the biological replicates. So what data we have, these are the biological replicates, and these are not the technical replicates. So technical replicates are those that the same sample has been run twice to generate 2 times the data to check the run to run variation. So but these two data cannot be taken as an individually for analysis, because the same sample. So you take the average. General procedure is that you take the average and consider it as a single sample and perform the analysis. So here these are the biological replicas where the same 7 control healthy controls are taken and the 7 patients are taken samples, and then it has been analyzed, then it is, the data has been acquired. So these are the two. So this is the biological replicates. The more number of biological replicates you have, the more robust your analysis becomes and more statistically significant it becomes. So there is a test called power test, statistical test, to determine that what number of samples are generally required for a particular variance you want to measure in your study. Then what are the number of samples that you need to study and analyze. So this is difference between a biological replicate and So am I clear, right? Everyone have moved to that portion in columns unpaired. Now we will choose the data file. So where have you kept the data file? Just choose it and then press the submit. Okay, so this is an online tool, so it will get second. Why this is happening? So there is some error is coming. Just check into it. Ah, so you just, if you all are getting the same error, just change it, make it as a condition proteinitis and the NC and the TC, like the tumor and the normal, so it will take. So so just upload it and let me know after you have up. Okay, everyone. Or you all can speak up also if you are facing any difficulty. You all could follow na what I did. Have you done till that the it has been uploaded in the meta-analyst? Okay, so we'll moving forward. So what you can see that here it has written a total of 9652 40.5% missing values were detected. So for this data set, so this uploaded data file contains 14 samples and 1702 proteins, and 40.5% of the missing values were detected. Means the data set that contains the 1702 protein in the 14 samples, across it 40.5% are the missing values. So I'll just paste this. So every one of you got the same result, right? So if you, so you can understand that from where you will understand that how many features are there. So the when it is written where it is been written 1702, that means these are the number of features. So these are the number of proteins that are present in your data set, and these many numbers of are there across the P cross N matrix. Okay, so now it is written that we need to perform the, since 40.5% there are missing values. So what we need to do is that we need to perform the missing value imputation. Now we need to specify that how many features we need to remove with greater than percentage of missing values. So can you all tell that what ideally what percentage we can provide here to get the percent, means to move to delete those features and impute the remaining ones. So what can we put here? Any guesses? Any number from every one of you? Yeah, anyone, any guess? You all have gone through the lecture, na? So we will keep it as 50%, or we will increase it or decrease it. Or we will not remove any feature. No one, would we? Okay, so generally uh, we can missing values as 50% also, but generally what we do is that it will, this meaning is that with those features that are present in 50% of the samples, it will be imputed, but which where it is more than 50% is missing values is will be removed. But we generally keep do it as a 30, so that a particular feature should be present in 70% of the samples. Means a particular feature should be present, if there are 10 samples, it should be at least present in 7 samples, and then only the remaining 3 samples will be imputed. Otherwise the samples will be, otherwise the feature will be uh, taken out from the analysis. So if we give 50%, it will be 5 samples, but if here we are giving greater than that, wherever the feature is, any feature is present, is missing from greater than 30% of the values, so it will be removed. So generally it is taken here. So different studies can take different, but it it cannot be more than 50. 50 is the maximum, and general norm is to take 30%. So am I clear, right? So please let me know if you are not understanding or thing. So there are different way we can take and understand that how you can perform the missing value. So here we will do KNN feature wise, or the you can do by the different samples, sample wise, or PPC, or the different kinds of, but we generally use KNN using feature wise. So it will check on the features and it will perform the impute. Sample wise means it will check on the samples, but we need the features to be imputed. So here we are taking just KNN in feature wise. So I'm just pasting it here. So all of you are getting the same results at the same page, right? Please let me know if you all face any difficulty or any. Now we will process it. Correct? So now after processing, and now we need to filter it, because this works in a uh, open access, in a software, means in a database feature. If there are many features, it will remove those, and which are present in the, between the interquartile range, it takes those only. So we will just here give as proceed. So now here comes that which normalization you need to perform. So everyone is at the same page where you need to perform the normalization. So just quickly respond, please respond, otherwise I'm not getting that you all are understanding or not. Urvi, Jaseda, Sukanya, just, yeah, fine. So now, so what normalization we should do? So you all are familiar, right? Normalization, what is transformation, what is scaling? Yes or no? Yes. So what normalization should be used, should we, and why? Should we use quantile normalization here? Yes, any response? Generally, quantile normalization will make all your data come at a range, and you cannot differentiate between, so it takes two different distributions, that it will likely to make the as identical, which will not help you in understanding the differences, whereas some datasets can use quantile normalization, but generally when the feature numbers are less, generally it is not used, and it is used as a median normalization, followed by log, and the transformation is generally taken as a log transformation, so that you can compare the different classes, and finally if you want to scale it for the, and do for the means centering or the scaling. So but we will, firstly we will do is that normalization by median and log transformation. So these are the basic two things that is being done to normalize your samples, so that you can compare the two samples to different groups analysis. So now we will just, before that, okay, so now we will process it, we will normalize it. Now we will go and check the view result. So we will go to sample view and check. So it was like before normalization, you can see the distribution and the box plots, and this has been done after normalization. It is mostly normalized with a kind of Gaussian distribution. Since it is a real data, we cannot get a fair normalized distribution, and this is the box plot where most of the median lines of each of the samples are in a particular line, so that we can compare the two different samples. So everyone has so the quantile normalization is a test. In this normalization, what is done is that it takes the uh, maximum deviation value, and it makes them come, takes the average of those values, and makes, means all the features are generally made come closer of both the groups, so that so for that, after quantile normalization, when you try to compare the control versus disease group, you don't get a quite a fair of the fold change difference, because it generally tries to make it in a same scale. Okay, so generally that's why it is not favorable, and it is not used for the normalization techniques. and when the number of features are especially less, it generally it does not take in. We generally perform median normalization only for the proteomics data. So all of you are clear that how you will check that it is coming before normalization, how the intensities are is looking, and after normalization how it is looking. And the most important thing that you should look that the samples, all the samples median line should come near about at the same place, so that you can understand now that you can compare these two differences samples, and that will not be a technical variation but are due to a biological variation. So everyone has got the result and understood. Okay, so just so this is before normalization and after normalization. Okay, so now we will proceed. So now you can see that there has been univariate analysis, uh, significance analysis, various things are there. Now firstly if we look at the fold change analysis, we know that we need to find out that what are the proteins that have come fold change threshold. So now at least twofold difference, so you can go and understand that the group means, what are the group means in the control. So you can select here NC versus TC or TC versus NC. So means TC versus NC means the tumor versus the normal. If you check and fold change 2, so you can see that there has been significantly upregulated proteins that have a fold change of more than 214 in the disease state, and the downregulated is 227. Okay, fine. So everyone can understand that how you can get identify those proteins. Download this Excel file, so you can see that these are the protein name, these are the fold change, and these are the log to fold change. So here you can go and identify, but these are not we haven't done yet a t-test to identify that these that fold change that are coming, they what are the p-values of those fold change. So now what we will do is that we will go to identify a t-test. So group variance we will keep as unequal, and 0.05 FDR is giving only 4 significant tests. So here the data was looking fairly normalized, but still if you don't want to be dependent on the distribution of the data distribution, that it is Gaussian distribution or not, you can go for the non-parametric test and give. Say here is this 19 proteins are coming as significant in a raw value of FDR of 0.05, means less than 0.05 FDR. So now if you want to increase and make it little non-stringent, so here you can see that 171 significant proteins are coming, but here can have some chances of false positives. But if there you give as an FDR or the adjusted p values, then you are definitely, it will not be the corrections are there, and it generally are absolutely biological variations that you can see observed in your data sets. So you all understood, right? You all have gone till this place, right? Now, so you all have seen in the different papers that volcano plot is there. So now you can see that you can also draw a volcano plot, and you can also have analyzed the data and can understood the data. So what are being generally reported in different papers. So you can see that how you can build and come up to this place. So here you need to find out that what are the fold change you want to give. The direction of comparison, we generally it is done is that disease versus control and not the control versus disease. So if we go for the uh, non-parametric test, group variance unequal, and you submit it. So you can see that in this volcano plot there is significant upregulated proteins in the disease state is 50, significant down regulated is 100, and the volcano plot will consider both of your log to fold change and your p values that you have gotten from the non-parametric t-test. So here you can see that how easily you could build up that volcano plot that you were seeing in the papers of the papers like in the CPTAG papers they have also mentioned about this volcano plot. So how easily you could build, so everyone could build it, right? The volcano plot, correct, right? So everyone could, yeah, so everyone has reached to that place. So now if we go for the correlations, that the sample to sample correlations, so we will choose the samples Pearson R. So color contrast you can give and you submit it, right? So here you can see control to disease. So mostly in the biological samples, generally the samples need, because the biological samples mostly it remains the same. So the correlation generally should come above 0.7. So here we can see it is coming around points above 0.7 with the correlation, but there are some samples which are showing a very not so good correlation and only within the range of 0.2 or 0.3 correlation. So we can check on these samples that why they are showing this and find and can remove those, or we can take ahead. But generally for the biological samples the correlation should come above 0.7 to check on that the sample qualities and the dataset qualities are good. So this is correlation. So another plot you have seen in the paper in the case study discussion what we have done is the PCA plots or the PLSDA plots. So PCA plots, I think in the next you can understand that it will come that different unsupervised clustering. So here you can see that the two different cohorts are coming in this case. Also the PCA plots, for example, you have known about the batch effect and all. So the PCA plots are also very much useful and fruitful in understanding that your samples have batch effect or not. So if you have samples run in certain batch, another samples in certain batch, and you do a PCA and you see that there are two different distinct clusters, so you can understand that there exists a batch effect and you need to perform the batch correction. So that PCA is also helpful in understanding the batch effects, but it is generally used to understand that how the two cohorts are different, your normal or diseased or condition. So you did another, you have all seen that the PLSDA. So PLSDA is a supervised way of clustering. It takes 70% of data is trained that this is belongs to this cohort and this belongs to this cohort, and after that the data is uh, the others are predicted. So here you can see that how usually you can see that this is the normal, this is the tumor, so there exists a difference. So it gives us the idea that there are some proteins that is causing the differences and we need to collect those proteins, and finally we can assess the biomarker analysis. So everyone is clear till this part. So so you have seen that there are so many plots that looks in the is being regularly used in different paper, and how easily you could come to this just the thing is that you know the concept and you have the data, and you can easily go step by step and can analyze those data and can come to a conclusion. So would we, Jasera and all, are you clear? So you all have understood, right? Do you have any question regarding it? So PLSDA plot is that the PLSDA is a supervised way of clustering where it trains on the data that this data belongs to this cohort for a certain percentage of samples, and after that other samples are predicted. So this is also mostly a classification technique. So here you can see that with a certain percentage of the proteins are making it as means it is a difference in the both the cohorts, that these proteins are responsible for this. Like if you see the important features, so here if you go, this protein is highly expressed in tumor and low expressed in normal. So this protein is responsible, or its intensity level is has a significant difference in tumor cohort and the normal cohort, and hence it can be a biomarker that is, or a taking a specially role in the disease, in this type of disease which is driving the disease condition. So these are the interesting proteins, or these are differentiating, and that's why we are seeing that we can see that there are two different cohorts of the two different clusters in this part, in this plot. So are you clear, Jasera? Okay, so these are the plots. So I think clustering and the things will be mostly taught in the next class. So week, this week was mainly focusing on the data normalization. So you can understood that how you can perform a normalization and how data looks before normalization, after normalization. So generally, the data should come in a Gaussian distribution shape, but the real data doesn't come always in those shapes. So generally we need to compromise on those effect, and what kind of normalization to be used, and the centering and the scaling to be used, and then we need to understand, and we need to perform the statistical analysis along with giving the corrections. And so more stringent we get, uh, more confident biomarkers we get, but generally also it doesn't, might get, we might lose some of the proteins or the signals. So this is for the metabo analysis part. So I'm just do you have any other question for this week, whole lecture on today's session, or anything? Does anyone have any question? No, right? Okay, so okay. Another thing is that what we are telling each week, that the different tools that you can take part and analyze for the proteogenomics approach. So if you all remember that previous week we had told that BWA or Bowtie2 generally helps in the alignment of your genome, of the disease genome with a reference genome or with a matched control genome. So now after the alignment, how will you view the alignment score? Like the MSA, multiple sequence alignment, if you all have done in basic bioinformatics, so you can see that there are sequences lined up, and you can easily identify that where there is a difference in the star mark, means where is a difference in the codon sequences, means the DNA sequences. So here for the this kind of human data, human genomics data, so integrative genomics viewer, which gives you a chromosome wise alignment score. So you all can try to read about it and try to use it. So this is it is also present in a web application, but you need to give a alignment file, a PAM file or SAM file. So after you visualize this, another step would be to then the alignment quality check will come. So I'll tell about the tools in the next. So this is an integrative genomics viewer which you can visualize the genomic alignments. Okay. So that's it. So that's it from my side for today. So do you have all have any question? Any question related to this week? Yeah, deep and shallow dilation. I'm coming it. Other any other question from this session? Okay, so I thought we were correct. So deep and shallow deletion would be generally what it is saying is that this is deep deletion refers to the homozygous deletion, or the shallow deletion refers to the heterozygous deletion. So if you go to this site, one second, I'll show you. So here they have mentioned about it. Okay. So okay, so minus to a deep deletion or possibly a homozygous deletion, and minus one or shallow deletion indicates the possibly a heterozygous deletion. Okay, so this has been mentioned in the C bioportals thing, so you can go more to it and understand, right? Homozygous means in the same allele, the deletions. Okay, and that means in the different alleles. So if not any question, then we will end this session for today. Okay, and we will meet in the next session in the next week. Okay, so I am stop sharing and ending the. Okay, thank you all.